It's really not funny.
No, there's the thumbs down. That's what I was laughing at. Hello, everybody. Welcome to Q&A with A&V. I'm Vincent Racaniello, and joining me from New York, Amy Rosenfeld.
Hello.
The lights look great.
Yeah, I had them fixed.
I know. It's a great job. It's actually better than my office. So
I had
sorry,
those fixed.
sorry about the technical issues.
Well,
Hello, everybody. We have 231 people. We have 81 likes and one not dislike, and I don't know why, but, since I complain about it, I guess people want to give us a thumbs down. I just want to tell people that we have passed 96,000 subs on the YouTube channel. How cool is that? The Lex Friedman bump, I guess. Right?
It's great. Yeah, it's good.
And last week, after we finished, my office flooded, but it's all cleaned and set up again, as you can see. And hope it's supposed to rain tonight, Amy. Did you know that?
I did. I brought my umbrella.
Okay.
But
all right.
Last week it was not that much fun whitewater rafting home.
I'm sorry. Did you get really wet?
Yeah. I mean, there were 6 inches of water. As I said, I whitewater rafted home.
I'm sorry.
Right. It happens.
All right. Let's jump into the questions here. And this is really the first one I'm seeing from account owner. And I'm sure it's not the first. So if you posted one earlier, and I know a lot of you did, please post them again. Should humans bring all life to space, including bacteria and viruses? Well, it would automatically come with them, right, Amy?
Yes.
Because you have your microbiome and your virome, and you can't get rid of them. So, the astronauts bring it into space. They brought it to the moon. Can't avoid it.
Nope.
But yes. What do you think about vaccinated people who allow their children to play with unvaccinated families and who have had COVID in the past month or so, 3 weeks? Don't you think that's risky?
So the vaccinated people don't have COVID. The unvaccinated people have COVID, right? Is that
Apparently,
the question?
yes. And the vaccinated people
Why
are
is it
letting
risky?
because the kids are not vaccinated, I suppose, right?
I don't know. It's not clear.
I'm assuming, Charlotte, they're not vaccinated, in which case it's risky, yes. I would not do that, and I'm sure Daniel Griffin would not.
Right, but if your kids are vaccinated...
If you're vaccinated, then it should be fine, right?
Right. It's not... Yeah, this question is not clear to me.
You could restate it, Charlotte. Okay, Ronald wants to know. Infection requires susceptibility and permissiveness, so isn't reinfection always likely since S and P are unchanged, or is there some other mechanism that can prevent reinfection?
Well, that's what the immune system is for.
Exactly. Immune system is supposed to kick in and halt the infection. You're right, though. The cells are always susceptible and permissive, though. But antibodies and T cells and other things, too, kick in. All right, to listening one. What do you have an opinion on whether the mu or new variants can escape vaccines or monoclonals? It sounded like it's behaving quite differently, some say. Amy, you have any insight into mu?
Just that it's not really a problem because it's overrun by Delta.
I think in Colombia, it's about 75% of the last 2 weeks isolates.
I don't know. When I listened to In the Bubble with Slavitt, who had Fauci on today, he said mu was overrun by Delta. I guess he's not up to date.
Mu has a lot of the same spike changes and one additional spike change, and has some other changes like, as Amy and I were discussing today, a deletion of ORF3. What does ORF3 do, Amy?
It's an interferon antagonist.
Right. Which means it could technically be more virulent, right?
In the tissue culture cell, sure.
Yeah. We don't know about people. So as far as monoclonals go, it might, because it has a lot of the same, the E484K and N501Y.
Wait a minute. If it has the same, why would it escape vaccine and monoclonals? The other variants that have those variations do not.
Well, it depends what monoclonals you use, right? And
Well,
so
it's very hard to know. I don't really know. I don't think we've done the right experiments because everybody does it differently and everybody's using different things. Some use infectious virus, some use pseudotypes. I don't know. It's a
yeah.
mishmash.
So what Amy
So
is saying, there's no standardization of neutralization
so
assays. Yeah.
doesn't really mean anything to me if you come and say it's threefold less reduced. I don't know.
Yeah.
And somebody else says it's fivefold. Or somebody says I see nothing. I don't know what it means since there's no standardization of it. Is it in the context of infectious virus? Is it the context of pseudotype? Is it on vero cells? Is it on A549s that are producing ACE? Is it in lung cells? I don't know.
The key here to listening one is whether the vaccines still prevent severe disease and death. And for Delta, they do. And I think they will do for Mu. I think that's the bottom line. Isle of Man, only 38 deaths, 85,000 population, 90% of adults vaccinated. We are back to normal. Bar masking in hospital and care homes. It can be done. Congratulations. Yeah.
I want to go there.
Okay, I'll get you a ticket.
Okay.
Just tell me what to do with the mice and the nitrogen and the CO2.
Well, they're coming with me.
Shankrati said yesterday on Twiv, if we had 90% of the U.S. vaccinated, we wouldn't have any issues. So Isle of Man shows that. How about that? My vaxxed friend has been in bed with COVID for 8 days, not hospitalized, just out of breath, feverish, tired. Do you think he's shedding virus?
No.
We don't think so. Amy and I talk every day, and mostly we agree, not always. And we do agree that vaccinated people who get infected don't shed infectious virus.
Even though that's what I was told at the Rosh Hashanah table on Monday night, but the person who told me didn't really read the guidelines or the statement properly on the CDC website because that's not what it said. But that's okay.
Was that an expert that said that at the Rosh Hashanah table?
No.
No.
No. She does not have a PhD in microbiology.
Could rapid whole exome sequencing be at all practical to identify males with deleterious TLR7 mutations and provide them with extra treatments?
You don't need rapid whole exome sequencing.
What do you do?
You can just sequence across that. You can just amplify that region out and do a SNP test.
Right.
You don't need to sequence your whole genome. You don't even need to sequence it all. Just get a primer pair that can discern the wild type from the variant and call it a day. That's what we used to do. Why do you need to go so fancy? It's a waste of time.
Can you explain gain-of-function research? What's the purpose of such research? You want to take a snap at that, Amy?
Gain-of-function research is where you make an alteration in a protein and you give it, and you hope or you predict that it's going to confer a different function from what it had as wild type. Purpose of it is to understand the function of the protein, the wild type protein in a controlled system.
And
mechanistically how certain things occur. It's a genetic property. It's actually a genetic assay. It's been going on since we started screwing around with bacteriophage.
Bacteria, viruses, fungi, all organisms are subject to gain of function.
Yeah.
So it's not a bad word. It's good, but the press and some scientists have painted it as a bad thing.
But we should be honest. It started with two scientists, one at Harvard and one at Rutgers.
Right.
And maybe a 2.5 if you count the guy at Stanford.
The guy at Rutgers
I'm only giving him a 0.5.
And the guy at Rutgers is the one who called me a cult. Okay,
You
how about seeing booster third shot as a way to protect people while the unvaccinated get their infection? I don't know. I don't
I
see.
don't
We don't.
understand how that's going to protect people while the unvaccinated get their infection. How long are you going to wait for the unvaccinated to get their infection? From now until 10 years from now?
It could
It's a
take a
good
long
plan.
time,
That's
and
what
in the
I'm saying.
meantime,
That's a good
your
plan.
your booster immunity is likely to wane.
Well, it will wane. Just it's the whole property that we talked about last week when I sent the article that said from Israel that says, "Oh, boosters are stopping infection." And I said, couldn't they be more creative and tell the truth because they already played this card?
Indeed. All right. Charlotte is clarifying. My daughter allows my granddaughter to play with her friend whose family are on anti-vaccine, and their older daughter just had COVID. Not a good idea, Charlotte.
Well, it also depends on how old the granddaughter is and if she's been vaccinated. So if the granddaughter is 12 or above, what is it, 12 or above and been
Yeah,
vaccinated,
that's right.
I mean, if she's wearing a mask, it should be fine. Even if you don't wear a mask, it's kind of probably fine. But wear a mask and call it a day.
Are these Q&A sessions streamed on other platforms? No, but if you'd like, I could do that. I could stream it to restream and then stream it to wherever you want. But I'm only going to be on this one, chatting with everyone. So let me know. And this is very interesting. The video hasn't even started. How do we know if we like or dislike it today?
Well, we put that little envelope to our head and we say, do we like or dislike? That's what we do.
That's right. That's right. Okay, let's see. Are you ever going to have Ian Lipkin on to discuss his bout with long COVID? Do you think we should have him on, Amy?
I don't think he'll discuss that. I think he'll discuss other things that he's far more interested in.
Yeah, I agree. I totally agree.
And yeah, I mean, I think Ian has spending some time with Ian recently. He is really interesting. He has a lot of good stories and a lot of good information and sometimes very good ideas. Sometimes ideas I don't quite understand. But he could probably say the same for me. But yeah, I think, he would talk about something else.
I agree.
I think he should talk about Gideon, personally.
I agree with you. He probably would not find his long COVID particularly interesting. However, It would be fun if I did it with you and Ian, Amy. That would be fun.
Yeah, that would be fun. But we could talk about Gideon. We could talk about he could talk about his chronic fatigue stuff and other stuff, right?
Yep. Oh, yeah. He's got a lot to talk about.
Yeah.
Oh, this is for Amy. How many places in the U.S. have the equipment to do genome sequencing? Is there even enough sequencing being done to be able to say that Delta is what everyone is getting?
Everywhere can do genome sequencing.
Right.
Whether or not it's done like in my immediate vicinity, I could do it across the street at Ian's, or I could put it in an envelope, have a drop box, and have it taken to New Jersey and have it sequenced. Not a problem. Is there enough to be? Yeah, there is enough of being done to say what percentage of infections are due to Delta. Yes.
Yep. You don't have to sequence everyone to know that you use statistical procedures. Amy will laugh at this one. I've been isolating too long. Vincent is beginning to look cute.
That's cute.
Oh, I'm sorry. Something must be wrong. Go to the ER right now.
He's fine.
Do taking monoclonals interfere with the development of natural immune building following infection?
Of course.
Yeah, especially if you take them early, right? But if you take them after 3 weeks, probably not. But they're not going to help you either.
I was going to say, what are they going to do then? It's like expensive pee. It's like taking too much vitamin C and then urinating it out.
We have 261,000 views on the Lex Friedman interview. A number of people are saying they like it. I'm glad you liked it. It was very thoughtful. Because he gave me a lot of time so I could be thoughtful, slow, listen to him very carefully.
Gave you the questions ahead of time.
It gave me a list of them ahead of time, but I didn't really prepare for them. I don't really
It's
need
okay.
to.
The answers aren't the problem. It's the person who's delivering the questions. That's the problem.
Are you not a fan of Lex?
You know when he said how people could snuff out bullshit on the internet?
Yeah.
It's amazing that he has 1 million views.
Oh.
We should have one. His mom.
But you still advocated me going. You advocated that I go.
Well, for sure, but that has nothing to do with whether or not he's a bullshitter.
Yeah.
That has to do with marketing your brand. You have to learn to play the game. It's like I can write a grant on stuff I don't really believe in. And I've gotten funded for it in the past when I was a postdoc. But I prefer to do, I needed the money and I did other stuff with it.
What virologic assays would you like to see performed that could provide critical characterization of SARS-CoV-2? Go for it, Amy.
Well, I don't understand what critical characterization means exactly. I think that no matter what property you try to alter of the virus, you need to do a plaque assay and look at the production of infectious virus. Whether or not you make different changes in the spike protein to look at entry, encoding, polymerase changes, NSP14 deletions, blah, blah, blah. I think you need to do a lot more plaque assays. This RT is not working for me. But it's also not working for Eric Nierenberg either. Didn't I send you his tweet?
No, I don't think so. What did he say?
Yeah, I did. I sent his tweet to you and Daniel where he says we should pass a law against anyone who says that RNA copies is correlative to viral yield.
Oh, I thought you were writing that yourself.
No, it was Eric Nierenberg. He wrote it. Somebody, Bill Jackson, the EV68 guy, retweeted it. And then that's how I saw it. And I thought it was great. I thought it was really appropriate.
How many years
have we been saying? We've been saying it for 100 years. Yeah.
Yes. And we've been ignored, and now other people are just picking it up. It's fine.
Yeah, but it's fine. I don't have a problem with that.
Yeah, I agree. I think it's just we have to use the right words. Yep.
Well, yeah, where is it? Yeah, Eric Nierenberg. I would like a rule that you are not allowed to write that you compared viral load if you only looked at CT values.
I agree.
Profound thought.
I wonder who's done that for the past year.
Well, I wonder who's always quantitated viral reproduction with plaque assays.
Well, wasn't I on the phone today with a fact checker who couldn't understand why RT-PCR wasn't measuring viral growth?
Yes.
I'm sorry I missed the AMA today. I had to do other things, and I'm still not caught up for today. I have to edit TWIV, blah, blah, blah. But I'll be back next week.
We didn't do mice. We have to do mice, and the CO2 needs changing.
Holy cow, we didn't do mice.
No, we didn't do mice.
You want me to come in tomorrow and do them?
Well, I don't know. Yeah, maybe, and then we can do the CO2.
Okay, I'll come in tomorrow.
Well, you're going to the incubator.
No, I'll come in to do the mice and the CO2. It's fine.
Well, where are you going to record Daniel?
I don't know. We'll see. Don't worry about it.
No,
I was going to try at the incubator, but I don't think
it's fine. You can just go to the incubator. I like this. Amy needs to talk in a way we can all understand. Really?
It's not very nice, folks. I understand, Amy.
She complains consistently about me.
I think a lot of people here like the way Amy talks. What's your favorite enzyme? Oh, boy. That's a tough one. Because it's a toss-up between RNA-dependent RNA polymerase and reverse transcriptase. But I'd have to choose RNA-dependent RNA polymerase because the virus
really? It's not 2A?
No, I like RDRP, even though I never really worked on it. I still think it's amazing. Very cool.
I think 2A has a lot of interesting properties.
It does. We did work on 2A. And
yeah,
I suppose, and there were a lot of problems that we didn't solve. So one could imagine going back to them in another life, but that's for you to do, Amy.
It's okay. I have lots of questions. And maybe I will go, considering the fact we still don't understand what 2-murine purine does.
To
Bob's result.
aminopurine.
Yeah, Bob's result.
All right. This young lady, overjoyed, is fully vaxxed with long COVID. Many PCR-confirmed reinfections in a year. Immunity never lasted more than 95 days. Is the third shot okay? I don't know what it will get you, but you could try it, right?
Sure. I mean, she might be one of these unlucky people who never is able to mount a good immune response. Like I'm not able to mount a good immune response against measles. So I finally said, stop vaccinating me. You're going to give me measles.
Right.
This is also lovely. Elisa Grace. Amy looks irritated, bored, and annoyed. Really? You know how I feel? Did you know that this girl can just, I don't know, like must have like the ball, so you have the envelope, and then didn't Johnny Carson have like the magic ball like underneath a sheet?
I don't remember that.
Well,
was young.
Somebody had them. Yeah, well, I was even younger.
Amy
is
Amy.
Porn.
No, she's not annoyed. Amy's not annoyed. She's not bored. I tell you, she wouldn't be here if she were annoyed and bored.
And irritated.
And she's not irritated. This is Amy. Just get used to it. Oh, Leo Laporte, the tech guy. So I have to say, maybe some of you know that my inspiration for this week in virology was This Week in Tech, right? Leo Laporte. Yeah. And so early in the... so I did some shows on his network. I did...
was he with Mark Pelletier?
Yeah, Mark Pelletier, Futures in Biotech. I did... Kiki Sanford had a science show. I did that. And when the pandemic began, I was watching Leo on This Week in Google. And he was going over TWIV website and saying this is a good resource for the pandemic. And he just said, "Oh, I know Vincent." Just kind of flipped it off as if, wow, yeah, I know Vincent. Okay. Oh, studying virology. So I hope to do the live stream virology course this month. You know, I'm really trying hard. I ordered a bunch of stuff on the train home tonight to try and get the studio set up so I could live stream it from there. So I have to the end of September. No problem. Okay, when they test samples for SARS-CoV-2, do the labs need to be BSL-3?
No.
No, they don't. They throw the swab, they put the swab in your nose, and then they throw it into a tube, and then they inactivate it before they do the PCR, which is good. Otherwise, boy, that would be a bottleneck, right, if they had to do BSL-3.
Well, why would you ever have to do BSL-3? You take a swab and you put it in Trizol.
That's right. Trizol will...
basically phenylchloroform.
Yes,
it
will inactivate.
Phenylchloroform. So it
inactivate
everything. So I don't understand I don't understand why I would ever need a BSL-3.
Nope. Anyone measured antibody titers for virus vectors? Is the immune response to the vector a real problem?
Years ago, they measured antibody response to adenovirus vectors when all those people died at Duke and they had to stop the gene therapy trials.
Yeah. So, the Sputnik was designed with two different ad vectors to get around this issue if it were an issue. But with the J&J vaccine, it doesn't seem to be an issue, nor with the Chadox, which is multiple doses. So it doesn't seem to be a problem. Right, Amy?
Yeah, it doesn't seem to be a problem. But, we had all of that information from, in the late 90s or early 2000s when all those kids died at Duke?
Yes. Amy, we are the notorious A&V.
Okay.
Maybe we're even a cult.
I'm fine with that.
You've emphasized there's no evidence for transmission by vaccinated people. What kind of experiment could be done or data collected to prove or disprove? Amy's been saying this the whole night. You have to take a vaccinated person, and if they turn out PCR positive, so they've been infected, take some nasal swabs and look for infectious virus and see how much is there.
Yeah.
And if you have 10 plaque-forming units, they're not likely to be transmitting.
10 plaque forming units? Are you kidding me? That's
How much do
not
you think?
that's not an acceptable plaque count.
Amy, how many PFU do you think you need to shed in nasal secretions to transmit?
Well, what is it? That CT, you have to have a million copies?
A million RNA copies, yeah.
Yeah, but that's dependent upon what. So we don't really know because you don't even know what the infectious to defective particle ratio is. Although probably Raul knows that by now.
Is
Because
he
I think he has.
sorting it
I
out?
think he has. I think he has a grant to use DI particles like he did for polio and his DARPA stuff.
Got it.
So I bet you he probably does know. You can ask him.
Now, the other thing I would say is another way to look at transmission would be epidemiologically. You look at very carefully contact trace cases and see if you can identify transmission among vaccinated people. Like in a school, that would be perfect if all the students are vaccinated. I don't think Amy would like that as much, though. Amy wouldn't like that.
No, I don't like that because that's basically then leading into the Bayesian modeling that Shaman does, and I don't agree with models like that. So, nope, not acceptable.
Arguing with a Robert Malone fan, I tell him there's no ADE in reinfections with new variants, so we shouldn't expect ADE when vaccine-induced antibodies decline. Any suggestions?
Well, you don't see even better in reinfected people. You don't see more severe disease. You see less severe disease. If you had ADE, you'd have. It stands for antibody disease enhancement. Therefore, you would have
Antibody-dependent enhancement.
So therefore, if you had a reinfected person, that person should be more severely have more severe disease, like
that's
death.
Right. There's no evidence for ADE, yet people like Robert Malone keep saying.
He also calls himself the inventor of mRNA vaccines. So it's getting to that time of year. No, it is. It's he does or whatever.
Oh, Nobel Prize time?
Yeah, it's getting to Nobel Prize time. And I wonder if they do the mRNA vaccines, if they will do the right thing and give it to the lady and the guy at Penn.
Oh, that would be the right thing to do. But
right. But they very rarely do the right thing.
Yeah, but it also depends on in vitro transcription, right? So
but didn't they already give people the prize for polymerases?
They did.
Isn't that what Letterberg's prize is for or something?
I don't know about Letterberg, but yes, I think that's correct. Okay.
So, yeah, I don't have a problem if they don't do it to the in vitro, that T7 RNA polymerase isolation and stuff. I will have a problem if they give it to like Moderna, CEO of Moderna or Pfizer, because it's really, or like Barney Graham or Kosmikia, because it's really those two people at Penn.
Yeah, they should get it. No CEOs, absolutely not. Please.
But it's I would be even more appalled if it went to Barney Graham at Kosmikia.
No, they should not get it. All right. Much as I like them, if you get over 100,000 subs, you get a gift from YouTube. That's right. But my gift is right here tonight. All of you folks coming here, 650 of you coming here every Wednesday to listen to us. That is our gift. Wouldn't you say that, Amy?
Yeah, I think it's great.
But yeah, with the YouTube, they'll give some kind of a thingy. I don't know what it is. It's an icon of their tube. Yeah, fine. That's good. I'm just happy to have a lot of people listening. Since microRNA regulates expression of immune genes, could SARS-CoV-2 dysregulate the microRNAs regulating the immune system?
In what context is this dysregulation?
Oh, like somehow antagonize the microRNAs that are regulating the immune system.
So you mean like that the RNA would act as a sponge?
It could be a sponge, yeah. Let's say a microRNA is needed for immune gene expression, and yeah, if you sponged it out on a circular RNA, you could,
Well,
repress
there
it.
isn't any circular RNAs that have been identified to be coming from the SARS-CoV-2 genome.
Yeah. I mean, it's theoretically possible, but we don't know of any
Well,
mechanisms. Yeah.
right, and actually microRNAs, the 99.9% of them go in the opposite direction of what you said. The only one I know of that productively regulates protein synthesis is miR-122 for HCV.
Right.
All the others are inhibitory, at least that I know of.
In the case of Nipah virus, the Hendra Nipah vaccine can't have been extensively tested due to limited outbreaks. What side effects with the vaccine and how effective is it? This is a very interesting story. You should go back and listen to my TWIV with Linfa Wang and
Was it Unchun?
maybe it was there was another. Yeah, Unchun, right? Unchun and Linfa. They were both in that episode, right?
Yeah, this is one of her really big interests.
So
and I thought it was a horse.
So
that was to the horse.
The Hendra vaccine was made first for horses. It's a spike gene vectored. I don't know what the vector was. I forgot. VSV or adeno. I don't remember. And it turns out to protect against Nipah. The same spike will protect against Nipah virus. So that is being trialed. Phase 1. It's already in a phase 1 in people funded by CEPI, the nonprofit. Mm-hmm. And then, so that will give you good safety data, and then they will continue with increasing numbers. So I don't see why it would be an issue. You never know, right? But I think it's going to be properly tested.
Yeah, you never know. But I think also, didn't I just see that there's like a Nipah outbreak in the place that Dixon has been, Kerala or whatever?
Kerala, yeah. That happens often.
Yeah, I think I just saw it the other day.
It's a part of India, yeah, Kerala, which is...
but it's not part of the Nipah belt.
It is not, right. Belt. And in fact, yeah, a boy died over the weekend.
Yeah, I believe I just saw it.
So that is unusual because it's not the Nipah belt, and they're not sure. There's, it doesn't seem to be associated with date palm sap, so they're not sure what the risk factors are, but that was talked about at the meeting.
Well, that's all of Ian's paper. That's the paper that nobody read but me. And then everybody focused on, oh, wow, they put tracking devices in the bats. And I had to say, but the point of the paper is, remember that?
How can I forget, Amy? You give me hell about it from time to time. For sure.
I do.
The thing about Amy, folks, if you do something wrong, man, you hear it until you say, it doesn't matter what you say.
Well, it does. You just can't rationalize it away. Don't ever do it again. And you cannot rationalize it away as, well, blah, blah, blah. No, it's not, oh, well, blah, blah, blah.
No, I understand. However,
you cannot
folks,
rationalize it away, and then you have to improve upon the next time. Don't repeat the same mistake.
The thing is, folks, Amy is a gem of knowledge and logic, so she is worth having those other issues with for sure. I learned very much from Amy. Which, with the other 4 coronas in circulation, would they have had similar symptoms and features when humans first got infected with them? Well, that's what Ralph Barrick thinks. He said that first way back in February 2020 when he was on TWIV the first time in the pandemic. He thinks, yeah, they might have caused the pandemic many, many hundreds and thousands of years ago.
I just sent you the article. There's a bioarchive article over the weekend that talks about the origin of 229E. And it's not exactly what we thought it was.
Oh, what is it? What's the origin?
Or what they hypothesize. Oh, God, do you want me to find it?
You can...
I'll find it. First of all, I have to find you. Then I have to push the right button. All right, so... Was it wildlife in Cameroon? Might be wildlife in Cameroon. Yeah, it's wildlife in Cameroon harbors diverse coronaviruses, including many isolates related to the human coronavirus 229E. I think you guys should do it.
Okay, we'll do that.
I think it's really cool.
Yeah, sounds cool. I love wildlife sampling. Not eating them, but sampling them for viruses, folks.
But I don't think I'm not sure it was I'm not sure that they, at the end, No, it's like the closest relatives in the camels, the bats for envelope matrix and RDP, blah blah blah. And then there was this really interesting paper that I sent you this morning about how they think like uh reassortment or recombination occurred in the pangolin
Mm.
for 229 for
Yeah,
SARS-CoV-2
yeah.
or something. Yeah.
Okay, we'll have to do that one. Recent rise in infections in Singapore seem to be highly vaccinated, something to be concerned about. What is the fraction of vaccinated in Singapore? If it's 80%, that's a lot of people who are still susceptible. So, no, it's not a concern to me. Is it a
Well,
concern to you?
well, infection and disease are not the same thing. So I'm not
Yeah.
concerned about infections. I'm concerned about disease.
Agreed. Okay. There's one here. Neuro. Okay. The neuro talk. I presume you're referring to Kieran Thacker on Neuro COVID. Good to see they don't see virus in the brain, but is there any guess what causes the neuro symptoms? Well, as she said, she thinks a lot of it is due to hypoxia because you can't breathe. You don't get enough oxygen. It just messes up your brain. And then probably inflammatory reactions. That's what she said, right, Amy?
Yeah. I think Carlos might agree with that.
Is it unhealthy to keep SARS-CoV-2 antibody levels high for a prolonged period as the boosters would continually do? Assuming you got a booster every 8 months. What do you
I
think,
don't know.
Amy?
It sounds like there's a potential for, kind of slightly an autoimmune syndrome or something. I mean, there has to be a reason why they naturally wane for us, right? And we don't keep
Yes.
them completely high.
Agreed.
So there obviously is something that we don't understand.
Agreed. Okay, Frank wants to know what biologically or chemically makes lick a stick difficult?
Well, I can tell him directly since I'm about to write a grant on this, right?
Well, for a different virus, right?
Yeah, but it's all the same principle, right?
Yeah, of course.
Is you have to have an extraordinarily high affinity, high avidity antibody, and they're very hard to find. And then you have to be able to make them as hybridoma from a hybridoma, right?
Yeah.
In mass
Yeah.
production. And so when a certain person said he had a box of Lick-a-Sticks in his office 3 months into the pandemic in the United States, everybody should have said, not possible. Because there was no way we could have found that antibody that quickly.
I'm not sure we even have it now.
No, I don't believe we do. And
So
because the truth of the matter is only that one paper that you and I, when we did the TWIV together, talked about. You remember I was so excited about the Biacore machine, and that I wanted to go to what's his name, Barry Honing and Lawrence Shapiro, to do those kind of experiments for our antibodies.
Yes.
Right? Remember that? That's the only paper I've really seen that has done that for SARS-CoV, for a coronavirus antibody during the pandemic.
Yeah.
I'm not saying that they haven't done it. It's just that they don't publish those results.
I think the current antigen tests are using antibodies that are not very high affinity, and that's probably in part why they're not very good at picking up asymptomatic infections.
Right. And how fast was I to when we talked about this idea with Ian, I was like, this is what you need. This is why we're doing this. And somebody else didn't really quite get why I wanted the machine. Remember that?
I do. Okay, Amy, you have said that the Israeli waning immunity data is wrong, misinterpreted, but Fauci on Slavit seems convinced it is solid. Can you explain, please?
Yes, I listened to Fauci on Slavit, so I know what he said. Yes. So they said, oh, look, when you have very high antibodies, you inhibit infection. And I agree with that. That's fine. However, you can't continuously, that doesn't mean the vaccine is preventing transmission or infection, right? So if somebody is infected, naturally they'll have high antibodies. They too will not get infected, right?
Mm-hmm.
So it's just you have a high antibody level and that blocks infection. But who cares when you go? It's like when I come out of the beauty parlor, my hair is perfect, right? And then 2 weeks later, it's less perfect. And then 4 weeks later, it's less perfect. And then 8 months later, it's not perfect at all, right? And I have to look good. It's true. You have to look good at 8 months later. You can't be going to the beauty parlor just spending $700 every 2 weeks. Like, that's like ludicrous. It's the same analogy. So when you test, when the antibodies are first induced, yes, you are protected. And then when you let nature take its course, immunity wanes, and you come and you say, the vaccine is no longer protecting against transmission. That's not the correct interpretation of the data.
Did you say that Fauci claimed that protection against severe disease is waning?
No, he didn't.
He didn't claim that.
He didn't claim that. He said mild to asymptomatic to mild disease is waning. And then he said that there's no antibodies in the upper respiratory tract. There's only antibodies in the lungs. And being that we work on a plethora of respiratory viruses, right?
Yeah.
And I'm looking at antibodies to my plethora of respiratory viruses. I'm not buying that we don't have any antibodies in our nose.
So Crotty said the same thing yesterday on Twiv. You'll hear him say that. He said that the upper tract is not protected. He said the upper tract is not protected as antibodies decline, but the lower tract is, because that's what you need to protect against severe disease and death.
Okay. But if I remember correctly, from Paul and Theodore's, one of their intermediate papers, which I thought was really important, but you glossed over and decided that was uninteresting, was where they looked at dimer IgA. Okay. And I believe that dimer IgA is found in the upper respiratory tract and the mucosal lining.
It is, absolutely.
Okay. And I believe that the conclusion, and Ian would agree with me, that the conclusion is the vaccines all need to induce dimer IgA, not IgG.
Got it.
And because you do it IgG, you do through the vaccine because it's systemically given. But I bet you if we took FluMist or whatever the nasal vaccine is for flu, and we assayed whether or not there was IgA in people who got the inhaled version of the flu vaccine, I bet you we would find IgA in the nasal cavity, the oral pharynx, all the way down to the lungs. And then over time, it automatically decays. Right?
Yes, agreed.
Okay. So what was Shane's point again then? Because
he says as antibodies wane,
which antibodies? IgG, IgA, IgM?
Yeah, well, he didn't specify. So it's a good point. He just said they tend to wane first in the upper tract, which is why you get the mild infections, but they remain in the lower tract. But it's really memory. It's not the antibodies. It's the memory cells. Maybe he said the memory cells leave the upper mucosa sooner than the lower tract. We'll have to listen to it to remind us.
Well, that could be true, but do you know of any memory B cells that make IgA and not IgG?
Shouldn't there be memory B cells?
I don't know. I'm not an immunologist. That's my question.
I don't know, but I think there should be, yeah.
But the majority of time when you're talking about memory B cells, you're talking about IgG.
That's true, yes.
And this was a caveat to when we wrote the Ben grant, right? And then I wanted to build it into the grant that we just wrote with Ian, right? That we were
correct,
right? Didn't I say that we were sacking the mice temporally to look at the IgA, IgM, and IgG responses, right?
Yes.
That was actually, I said that would be a focus of the next project because this project is already too big.
Study proposes TLR7 variants in severe COVID males. How can this mutation of TLR7 deleterious and why in males? Well, TLR7 is X-linked, and males only have one copy.
Yes.
And TLR7 is a risk factor. Yes, that was shown a while ago, right, by Casanova?
Yes.
So it's the TLR7 encodes a sensor. So if you don't sense infection well, you're not going to respond well to it.
Yeah.
So women have 2X chromosomes. Why aren't
Oh,
the
wow. It's 8:50. We have 3 more, and then I have to set my ELISA.
Okay, why aren't the makers of the mRNA vaccines updating with the Delta and Mu spike variants? So they are. However, Exchange said yesterday on Twiv, you don't need it because all you need is a boost. He said, for example, if you get infected and you recover, your body actually makes a wide range of memory B cells against all kinds of spike variants which are not even in you. And it's just a matter of teasing them out by vaccination, for example. And then you get an antibody repertoire that can neutralize all the variants as well as SARS-1 and MERS coronaviruses. So he says you don't need to boost with the variants. You just keep using the ancestral spike and that's good enough. It's an interesting idea, Amy, that the immune system makes a big range because of hypermutation, right? It makes all kinds of antibodies in anticipation of any variant that would arise. You just have to tease them out.
Yeah, I mean, I don't know why you guys are all that surprised it does it for everything. But this is the point that Paul, where Theodora said you didn't need a booster, and Paul was like, you need a booster to round out the immune response. I wasn't really quite getting it, buying it, but
that's the idea. We'll see if
Yeah.
that's true. I mean, they're doing it. Is there a website to look for local outbreaks of virus in my community? So
I think
your
there's
state.
a map part. Right?
What is it, Mamie?
I think every state has a map of the counties,
Yeah.
but I also thought it was on like the Hopkins site.
Yeah, your state should have a Department of Health that has local case counts, but the Johns Hopkins website, just look for Johns Hopkins COVID counter or something like that, should have it as well. All right, only one COVID question. What's the incidence of long COVID after a breakthrough infection? As likely as getting hit by a meteor while crossing the street? So Daniel says we don't have the data yet. He did a paper last week which said that after 28 days, about 50% of the vaccinated people with breakthroughs have still symptoms at 28 days after recovery, to which I said, it's not really long, is it? He said he agreed. Yeah, we have to look out longer, so we don't know. In that one small study, 50%. But I think
thought that by 60, I thought that there was like something about 60 days or something.
oh, there's another study at 60 days? Yeah, I don't know.
I don't know. Maybe I'm getting confused. I don't know. But the lady who is running the Survivor or whatever website, the organization for long-term COVID, she thinks that you can get long-term COVID after a breakthrough infection. Even
I don't
if
know.
vaccinated.
I just don't know that we know enough to be able to say that because we're not far enough out from vaccination and breakthroughs
But
are
I'm
relatively
also
recent.
not clear. I'm also not clear that. So when you test positive after a vaccine and you have quote unquote a breakthrough infection, I'm not. And even if you develop symptoms are usually after the peak of viral replication. I'm not sure that it's not an aborted infection and the fragments of the RNA and the proteins are triggering all the immune responses that are leading to the cytokines, which is why you get mild symptoms. So I'm not even clear that a breakthrough infection is actually ever going to make a productive protein. I'm a little, I don't think
Agreed.
we have enough information.
No, I agree. No, we don't. We certainly don't on long COVID after these. But we
Right,
will at
but I
some
don't
point.
even think we have enough information to know whether or not, So from the Singapore data, you would say it's an aborted infection. It's
Right.
cleared rather rapidly, right?
Right,
Within 3
right.
days. That one piece that they're amplifying for the QT-PCR is cleared within 3 days.
Yes. Unlikely to be a productive infection. I agree. Stay
So
tuned.
I'm not really clear. All right. Do you want to do one more or what?
Yeah, one more after this, Jason. Thank you for your super chat. Appreciated. And then Buddy Groom wants to know if antibody level isn't indicative of strength of immunity, why are boosters recommended? How is the effectiveness measured?
Effectiveness of what? The vaccine? It's solely measured on whether or not you get some level of antibodies, right, in your serum. And how they quantitate those antibodies, again, is unclear to me because none of this is standard.
Yeah, I mean, the vaccines are tested to prevent disease of some sort. And as antibodies decline, you may get infected and get mild disease, but you're protected against severe disease, which may be a function of T cells and not the antibodies that are actually left in you. So we really don't know what protects you. People think it's antibodies, but we're not sure yet. All right, Amy, thank you so much.
All right. I'll talk to you tomorrow and decide what to do about the mice and CO2.
Okay. Good night. Thank you
All
very
right.
much.
Good night.
Bye-bye. All right, folks. Let's see. Oh, is there data? Are there data comparing a symptomatic disease in people with natural versus vaccine-induced? Yes, there are, and they are similar. Depending on the study, one will be slightly better than the other. The key is that people who are infected and recover, they have heterogeneous immunity. It's spotty, right? Some people have good immunity, others have moderate, others have poor. And you don't know who they are. And that's why we say if you've recovered, still get a dose of vaccine. Because then, of course, your immunity is phenomenal. It's better than anybody else. You can neutralize all variants and you can handle SARS-1 and MERS coronaviruses. So although on average the immunity after infection versus vaccination is similar, and Shane Crotty talks about that on Twiv, which is dropping hopefully at midnight if I can edit it tonight. It's heterogeneous. Yeah. Thank you, Morris, for your contribution. I'm glad you liked the Lex podcast. So I appreciate having a lot of time, and I think he asked thoughtful questions and let me talk, right? All right, you have to take a different vaccine after one dose of Pfizer gave you a reaction. My allergist wants me to take 40 mg of prednisone for 4 days. I can't answer that. We have to have Daniel Griffin answer that. I'm pretty sure he has in the past. But let me check with him. Shoot me an email at vincent@microbe.tv and I'll let you know what he says. Recently studied by Paul B. Nash, individuals with previous COVID plus mRNA have really high levels. Yes, that's what we've been saying for weeks now. It's several studies. His is one, and he was on Twiv recently. Yes, so natural infection seems to prime you to make all kinds of antibodies against all kinds of spikes that you may not even seen yet. And then the boost brings those out. So now you can take care of any variant. And so that's why we don't use variant-specific boosters. Just a regular ancestral virus booster is enough. My uncle suffered from late-stage Alzheimer's, had COVID before dying on Monday. My condolences, Richard. I'm sorry to hear that. How are deaths with multiple causes tallied? Does COVID aggravate Alzheimer's? Well, it could if you're hypoxic, right? And you have your brain is being starved of oxygen. That would make it worse because we know in people without Alzheimer's it does that. So it depends, right? If in some cases you have a diagnosis and the death is caused by COVID, but not in all cases, and that's where we do excess mortality, right? Where you take a baseline number of deaths in any particular country, and then if you have a pandemic or an epidemic, you see how far you exceed that, and that's a good estimate of what is being caused by the virus, either influenza or SARS-CoV-2. So that's because you don't always have COVID on the death certificate, right, for cause of death. It depends on the doctor. Do endogenous microRNAs inhibit viral replication? They can. There are some examples, yes, of cellular microRNAs that can inhibit, but they're not not numerous. And there are also some viruses that encode microRNAs that antagonize or regulate host processes as well. Yes. Does a lower inoculum in vaccinated people give B cells more time to respond? So I assume you mean, Michael, you're vaccinated and then you get some virus in you, small amount, right? And so, yes, you would have more time, because assuming the number of PFU you get is above this threshold to start infecting cells, right? Because if you have too low a number, it'll be taken care of by other defenses, chemical defenses, and maybe innate immunity. If it's above that and there are just a few cells infected initially, yeah, it will give more time for the memory response to kick in, as opposed to if you get a big dose of virus and many, many cells are infected initially. There you may have a more of a symptomatic infection. I mean, I'm speculating, right? Because we don't have data on this, but that's what's logical to me. You have answered in the past. My question is, does cigarette smoke carry virus, and can someone be infected from smoke? No. You can smell cigarette smoke long distances, right? Because they're organics, not large particles, but they do not carry viruses, no. That has been looked at before. If someone unvaccinated gets monoclonals, isn't that the same thing as a vaccine? No, it's not. So a vaccine is a modified form of the virus, which is given to you that induces you to produce antibodies and T cells. And then you have memory in the form of memory B cells, the ones that make antibodies, and memory T cells. When you get monoclonals, you're getting antibodies, already made antibodies, which can, halt the infection. But you will not have memory because you're getting antibodies. You're not getting memory B cells. So it's not the same as a vaccine. It's called a passive vaccine because you're getting the product of the immune response, but you're not making memory, which would be an active vaccine. Thank you for what you do. You've been a helpful guide for me through the pandemic. I'm a pastor of a small church in West Virginia. Over 90% vaccination rate in our church. Congratulations to you, and good for you for listening to science. I'm very, very happy at that, and thank you for telling us. Richard, thank you for your contribution. All contributions, of course, go towards the incubator. The incubator, what's up with the incubator? That's our new studio in Manhattan, 2 blocks south of Penn Station. I have been unable to get there this week to work on it. And as you heard, I wanted to go tomorrow, but Amy has some things that I need to help her with, and that takes priority for now. I had hoped to record the Daniel clinical update there tomorrow evening, and I don't have a camera there. I don't have a recording device. I don't have a microphone, so I have to bring all that in tomorrow. So we'll see if I do it. But as I said, it's my goal to get the incubator running before the end of September so I can start live streaming the course there, because I'm thinking of doing it at 11 a.m. and Eastern time. So I'd like to do it there. I'd go right from home there. That would be awesome. When there's many epitopes on a single virus, and there usually always are many epitopes on any protein, does the immune system select for many of the epitopes or only a few? Well, there are. So not every part of the protein becomes an antibody epitope or a T cell epitope. Only certain parts, and the B cell and the T cell epitopes are different. And there are rules for what constitutes a B or a T cell epitope. But as the antibody response matures, right, and that happens in lymphoid organs in what are called germinal centers, where the antibody genes mutate and they make different antibodies. They could make antibodies to an epitope they didn't see, or an altered epitope, say, with 1 or 2 amino acid change, so you could be protected against future variants, say. So you don't have to make it against all the epitopes anyway, because they're not all important for protection. Okay, Slovakia. country in the EU. Yeah, I do know that, but I don't know every country. So thank you. Permitted vaccination of all children above 5-year-old with Pfizer, even without EMA approval. Would you recommend it, considering the FDA has approved this in the U.S. yet? Recommend it for what? For other countries? Every country has its own regulations and standards, and so I think you have to go by your own. So in this country, for example, the FDA would like you, if you're coming from a different country, to be vaccinated with an approved vaccine. If not, with their approved vaccine, one of the FDA approved, if not, you should get one of theirs. So whether it's scientifically correct or not, I'm not going to say. I suspect these vaccines will all turn out to be safe in kids, but I do want them to be tested first because we don't want to have issues, right? So let's be safe. So I don't think it's a good idea, no. Do we have data that relates the severity of COVID to how high antibodies or adaptive immunity would be? No, because, the antibodies and T cells in their first infection rise very late in infection when viral titers are already declining. So they really have very little impact on the outcome of that first disease. What really seems to be important is your innate immune response, your interferon response early on. If you have a poor response, you're more likely to have severe disease. If you have a good response, that contains virus reproduction, and you have milder disease. That's where the data are so far looking, but there's bound to be more for sure coming in the future. Is there any test? Here we have a different virus, that could distinguish between if a person has been naturally infected with varicella as opposed to having been vaccinated against chickenpox. This is a good, interesting question. Well, so I'm not sure. So varicella zoster is the name of the virus, VZV, varicella zoster virus, because varicella is chickenpox and zoster is shingles, right? So you could be vaccinated. You could get the chickenpox vaccine or you could get the shingles vaccine. And so you would like to know if you could distinguish if a person has been naturally infected with varicella, but you don't get first infected with varicella. You would get chickenpox first, right? Anyway, I don't think there's any test that can distinguish between the two. Not that I am aware of. Vincent, Amy, I just got my flu shot, my 10-year-old tetanus today, and one in each arm. How different or similar are bacterial vaccines to a viral? Well, there are a whole range of bacterial vaccines. They can be inactivated bacteria. They can be purified preparations from bacteria. They can even be attenuated bacteria, like the tuberculosis vaccine, BCG. No mRNA vaccines that I'm aware of, but that's brand new, right? So I'm sure we'll see some coming. And I'm going to go out on a limb saying there are no vectored bacterial vaccines. I could be wrong there. So I think the principles are the same, and eventually they'll come to the same point. Because there were a lot of, and still are a lot of, viral vaccines that are purified viruses disrupted in some way, as bacterial vaccines are, and many of the bacterial vaccines are purified as well. The tetanus vaccine, of course, is a toxoid. It's a protein inactivated, so it's not going to give you tetanus, and you make antibodies against it in T cells. Yeah. Oh, Kevin, you found us on Lex. Good. So Lex has a big reach, right? 1.5 million subs. I did it in part because I hope to get more people to our data, to our forum here. But I also thought it would be fun because, yes, good questions. And it was fun. Had a good time. Very efficient fella, it was really good. Okay, can you name a virus or two that have beneficial effects in a human's biology? Now, human is very hard, right? Because we can't do the experiments. We know in mouse models there are some examples of viruses that can be beneficial. And the one that I like is if you raise mice with it's a murine norovirus, basically. If you raise mice without bacteria, their guts are abnormal. And then if you infect them with a murine norovirus, it partially restores the defects. We don't have anything like that in people because we can't do those experiments. But we have a lot of viruses that are in us all the time. As we sit here, all of us have many viruses. They're not making us sick. They're probably beneficial, but we don't know how. Those include polyomaviruses and circoviruses. They're both DNA viruses, double and single-stranded DNA viruses. Almost 100% seropositivity in the world's population. People shed them. They're in the blood. They're in the blood supply. Probably beneficial, but can't prove it. Lots of examples in plants, though, of beneficial viruses that help plants be thermotolerant, tolerant to diseases of all sorts, heat, thermotolerant, I already said that. A lot of great examples of that, for sure. You think heterologous prime booths with 2 different mRNAs confers more robust response due to the different shells? I don't think so. I think they're very similar. There are heterologous combinations that seem to be doing well, like a vectored, an ad vectored, and an mRNA that look like they may be even better than the homologous, if you will. So we'll see about that. What long-term strategies would best protect those with compromised T cells? Well, that's a problem, right? Because the T cells are what protect you against severe disease. So the strategy you have to use is to protect yourself. You have to get tested often. I mean, you don't want to wear a mask forever. But while there's high circulation in your community, you should wear a mask if you're among a lot of people. You should get tested frequently. And if you're positive, go get monoclonal antibodies right away. And that's the strategy that'll save you. When news becomes a bit overwhelming, what do you do to de-stress? Well, you have to ask Amy what she does. So Amy's in the lab almost all the time. And often late at night when no one's around, and I think she loses herself in her experiments. I stay busy. I do a lot of communication, right? I do podcasting and writing and so forth. I do like a glass of wine with dinner, but I don't have wine. I don't have dinner before the live streams, and I don't have wine before a live stream because I need to be sharp. It can't be any less sharp than I already am. Question about flu vaccine in children under 12. Flu vaccine offers adults partial protection against COVID. Children already have a strong native immune system. Would it help them too? Well, it might, but it has to be the flu mist because that protection, it's only a few months, only works with flu mist. So it might, yes. And early in the pandemic, I advised people if they wanted, they could get an MMR, measles, mumps, rubella vaccine, which is an infectious attenuated vaccine because there's some transient protection against COVID, for sure. Mu was first seen in Brazil in May, did not displace Gamma, probably will not displace Delta. This is all a competition between fitness among viruses. It's what you have to remember. It's not just transmission, which many people think. And in some countries, in Colombia, mu is dominating, but it's not elsewhere. So it really depends on the competition, right? Lack of standardization really comes to fore with COVID. Is this something that might become an important topic at virology conferences? Is there hope for improvement? Well, if the conference covers clinical development, clinical assays, diagnostic tests, because that's where the standardization is important. And there are plenty of those. Next week, there's the European Society for Clinical Virology, and I'll be doing 2 TWIVs there, actually. It's all online, and they'll be shown later. I'm sure they're going to discuss this, for sure. Actually, I'm going to do on one of my sessions, I can bring it up. It's actually a good question. What makes some people super spreaders, and why do some unvaccinated people not get infected? Okay, so there are, so 80% of infections are transmitted by 20% of the infected people. So most people are not transmitting, actually. So that explains why the husband is infected and the wife doesn't get it. He is just not transmitting, and we don't understand why. It may have to do with levels of infectious virus, but since we rarely measure that in people, we don't have any data on it. And that's what I think also. So those 20% of people that transmit 80% of infections, I think they make a lot of virus. They shed a lot of virus, and that's why they do that. I don't think there are super spreaders. I don't call them super spreaders. There are super spreader events. So if one of those people, one of those 20% of people go to a crowded venue, then you have a super spreading event because they're shedding a lot of virus and there are a lot of people around, so they will infect 20, 30, 40 people or more. That's what I think is going on there. I think it's all about amount of infectious virus, but since we don't measure it very often, we don't know. Is it right to think that the mRNA vaccine risk is a subset of virus risk other than infection injury? Well, some of it may be, but it's always much less risk than what the virus does, right? So whatever the risk, whether it's clotting or myocarditis, the number of those are much lower than you get the same things as with COVID infection, but at much higher frequency. So yes, in a way, it could be that they are subsets based on spike or something like that. But really, the risk-benefit is clearly in favor of immunization, in my view. Now, people can have their own view. That's fine. I don't think it's correct, but you're entitled to that, right? So I got COVID, and I started all over. My dog can get COVID, so I'm just worried. Yeah, your dog can get COVID, so stay away from your dog while you're sick, right? For sure. They don't seem to get very sick, though, dogs and other animals. Okay. Well, sometimes I don't know if it's a comment or a question for others, so here we go. If attenuated vaccine provides a better immune response because all the viral proteins are present, why can't we add additional proteins to the mRNA technology? Yeah, you could, and that's being considered for sure. The question is which ones? The spike is the only one you need for making neutralizing antibodies, but for T cells, that's harder to know. There's certainly T cell epitopes in spike, and there's certainly T cell epitopes in other proteins, right? So the question is, which ones, and how do you, T cell assays are not easy. They're not clinically very useful. So that's going to be a hard vaccine to develop and test, frankly. I've heard vaccine manufacturers say that. That's why I'm repeating it to you. So I'm not sure we're going to see much more than spike for the immediate future, especially if it turns out that a boost really gives you the same kind of antibody breadth and affinity as infection followed by a single vaccination, then that's the solution. I just don't see the data yet, but I'm willing to look at it and accept it if true. Are vaccinated people transmitting? Yes, Eurosurveillance paper, they calculate effectiveness of transmission 71%. But again, these are epidemiological studies. It is one scenario, a household scenario, where it is easy to do that kind of contact tracing, right? But you can be fooled. So I don't know how real that number is. We need more for sure. Concerned about rising cases of moderate COVID. Where do you see these rising cases of moderate COVID? Do you see a publication or is it anecdotal data? You have to be very careful about anecdotal data. No, the boosters make perfect sense without tweaking because, as I said, your exposure to the spike alone in a vaccine is giving you a broad response to include variants that you haven't even seen that are not even encoded in the spike. Remember, infection followed by a single dose, you get a huge breadth of antibodies that handle any variant. So that infection gave you that ability. And probably vaccination followed by infection will do a similar thing, although we don't know the answer to that yet. Why are COVID deaths increasing in the UK? Because there's still people who are not vaccinated. And, even a small percent in a population of many millions is a lot of people. And if you look at them, I'll bet they are all unvaccinated with I would say, severe disease and death. The moderate disease has always occurred, even in vaccinated people. Should we be worried about people developing autoantibodies to ACE2 and interferon post-COVID? Can a vaccine potentially create the same antibodies through similar pathways? I don't see why you would be worried about autoantibodies to these proteins. It's not clear to me what the mechanism would be at all, so I do not worry about it. If SARS-CoV-2 is going to be endemic and most of us are going to be eventually exposed, would it be better to be exposed at the time of our own choosing and possible control? I think it's best to be exposed after being vaccinated. If you're not, you're playing roulette because you don't know when you're going to get a severe infection. And more importantly, even if it's a moderate severity infection, you might get long COVID and never get over it. So that's why I don't think it makes sense to be infected before you get vaccinated. How much does vax antibody response vary between people of the same age, health? So the vaccine responses are quite homogeneous compared with infection immune responses, which are quite heterogeneous. As I mentioned earlier, Shane Crotty talked about that. And that's been reflected in the clinical trials where they did, from 18 and up, a very homogeneous response with antibodies and T cells. Well, thank you, Rach, for the invitation of the Isle of Man. I'd love to go, for sure. Someday I'll be traveling again other than Austin. Actually, next year I'm going to Europe. Going to at least Zurich in April, for sure. Unless they cancel it. Do you think SARS-CoV-2 was circulating in the U.S. in December 2019? Did I see some serological data about that? I'm not sure if I did. I would want to base it on that, and I just don't remember. Of course, for January, for sure. I wouldn't be surprised December. It would be quite rare, and you'd have to find it in, banked blood or something like that. Could be done, for sure. Yep. But, I think it was likely circulating in China mid-October, early November, and travel is extensive. So sure, why wouldn't it be somewhere in the U.S. in December? Absolutely. Do you think Fauci was asked by the White House to publicly support boosters? I have no idea. Even if he doesn't support their use. I think he thinks they're needed. He thinks that although currently the vaccines protect against severe disease and death, he thinks it's going to wane in 3 months. That's what he's thinking. And so he wants to preempt the waning. All right. So I don't think it was the White House. I think that's the way he's thinking. Vincent, where does virology go in the future? Well, virology has gotten a big boost from this pandemic, right? In the sense that a lot of scientists have entered virology who were not virologists before. I don't know how many of them are going to stay. Funding for virology has not increased substantially, so to maintain that momentum of research, you need more money. I don't know if that's going to happen. NIH research support has always been poor for all science in the U.S. The budget is $37 billion. It's nothing for the kind of science that we do. It's attracted a lot of people to the field. A lot of people want to get PhDs and do virology research, which is good. So in other words, it's potentially expanding. And the ability to study a widespread infection in many ways, mainly immunological, those have been the best studies, I think, has given us a lot of information and will continue to do so for many years. So I think virology has a great future because of this impetus. But more importantly, we're going to keep having pandemics. and we need to be ready, and virology is going to be at the forefront of that. So I think we need to fund it properly. You don't need to fund me. I'm finished with doing virology research, but you have to fund labs that are doing it and fund them properly. Right now, it's a killer to get research support for your laboratory. Ask Amy about that. So that has to improve, otherwise it will die on the vine. Okay, J&J, what do you know about 2 doses? So a press release came out last week from J&J saying this giving a second dose of J&J really substantially boosts antibodies, as you would expect. But I'm waiting for the preprint on that. I haven't seen it yet. And so far it looks good, but it's only a press release. In the UK, they want to combine a third COVID with the flu shot. What effect would the adjuvant have on Chattox? It would be my immune system's third encounter with the same adenovirus. I don't think that the adjuvant would have any effect. Chattox is a virus, right, that is binding to cell receptors and getting into the cell. Doesn't care about adjuvant at all. So, but of course, I guess your flu vaccine is adjuvanted. Not all of them are here in the U.S. Was the second of the 2-shot Pfizer-Moderna regimen too soon? Yeah, it probably was, but we were trying to expedite it because we were in an emergency situation, right? It would have probably been best to wait months. But until that, so the problem is that in the clinical trials, until the second dose of the mRNA vaccines, you didn't have very much neutralizing antibody, and your protection was not in the 90s. And so the second dose did that. Now, whether you let the first dose mature more, whether you would have achieved that, I think you might have. So I think more time would be best. Yeah, at least a few months maybe. You know, many boosts are given months apart. So I'm not sure that these are particularly adapted to 3 and 4 week intervals. Whooping cough versus mRNA, why the guidelines require 2 weeks before the 2 vaccines while others can be taken together. Well, the ones that are taken together are tested in clinical trials to be taken together, that they're safe and that they're effective, right? That's the only reason that people decide, well, Tdap, right? Tetanus, diphtheria, acellular pertussis, and they've also combined polio vaccines with that. Someone thought, well, why don't we give kids one vaccine that has them all? It's a good idea because people don't have to keep going back to the ped. And so they tested it, and it works. And that's the reason why you see certain combinations and not others. That's all. You think a nasal spray will be available in 2022? No, I don't think so. I think at the best, at the end, the trials will be done. But it's a tough one because if cases decline, they may not have enough people to do a phase 3. And here's the other thing. Now we have vaccines that work. They're the standard of care. You can no longer have a placebo-controlled trial for a vaccine. It's not ethical to leave out a group and not give them anything. Harder to test a vaccine. What you need to know is what is the correlative protection. For example, what level of antibody, neutralizing antibody, do we need to protect you after vaccination? So then they could take a new vaccine and say, okay, it meets that level or it doesn't. And we're not there yet. So I think the further we get into this and the numbers decline, the harder it is to make new vaccines. How many doses were used in the initial series for polio when the vaccines were first introduced? Initially, you got 3 doses each, type 1, type 2, and type 3. And then they combined the 3, but they still had to give 3 doses because to cover all 3 serotypes, you need to give 3 separate inoculations because they interfere with each other when you combine them. But it's better to give them all together, I think, because it lessens vaccine-associated polio. Technically, Twitch forbids multi-platform, blah, blah, blah. Okay. Well, then Restream is forbidden. But a lot of people use Restream, but I don't think anyone reinforces it, right? I think Facebook doesn't want you to do it either. but a lot of people do it. When will the 21-22 version of the flu vaccine be available? Well, it should be. It's usually available by now. So it isn't. Let's see. 2021-22 flu vaccine. Let's see. So yes, we are making them. We have the strains. No delays in production. So they should be available now because they're even at the end of August. They start to be available. So not sure why you say that. Why can't we get rid of warts? Well, we can. We get rid of genital warts, right? Because those are the ones that can cause cancers. They're sexually transmitted, and they're all over the urogenital area. Okay, use your imagination, and so we vaccinate people with HPV, human papillomavirus vaccine, to get rid of them, to prevent them from forming, and it works. You don't get the warts, and you don't get cancer. However, all the other warts that you get on your fingers and on your feet, they could be prevented by a vaccine, but they don't kill you. They don't make you sick. They're an annoyance, right? And so companies are not going to go for it because nobody's going to take it. So unfortunately, you can't fund people to do that. What is the antigenic distance hypothesis? Heard about this in regards to sequential seasonal flu vaccinations. So it's another way of original antigenic sin. The first flu you see, either vaccination or by infection, that's the one that dominates your immune responses subsequently. So if I first got infected in 2009 with H1N1, when I get subsequent vaccines, I mainly make a response to that. That's what it is all about. And so it's a problem, right? You want the response to be to the vaccination, which is the current variant circulating. And so it's hard to get around that. So people are working on that. Okay. German opera singer. Thank you for your contribution. Could heterologous prime boost? So I answered that. No, I don't think so. They're too similar. Do they change biosafety level classification rules for pathogens after there is already widespread community spread? No, they don't. So West Nile was given a BSL-3 years ago because of experiments done actually to treat people with cancer with the virus, and they got neurological infections. But it shouldn't be a BSL-3 because it's spread by mosquitoes, right? But it's never been retracted. SARS-1 has never been retracted there's no vaccine, and so that's a reasonable thing. Will SARS-CoV-2 be changed? I don't think so. I mean, you have to petition CDC. You have to have a lot of voice to do that, and I don't know if there's enough people doing it to petition it. So it hasn't changed yet. Could change, of course. I've heard an increase of cases of young people 18 up ending in the hospital with blood clotting. After what? Infection, vaccination? I have not heard this. I'm always suspicious of anecdotal data. People say, oh, I heard this or I heard that. For example, here's a one close to home. I have a friend who was fully vaccinated, and he and his wife went to a flea market or something in New Jersey, and a couple of days later, he came down with COVID. He got sniffles and sore throat, so he was tested and he was positive. And his wife, 5 days later, gets symptomatic and tests positive. So he said, oh, obviously I transmitted to her. I said, no, not obviously. The incubation period goes from 1 to 12 days. So you had a short incubation period. She had a longer one. That's another explanation, right? So that's what I mean by anecdotal data. Original antigenic sin. There you go. I just told you what it is for influenza virus. The Nipah outbreak was described in the press as having the potential to become another pandemic. Well, I don't think so, because Nipah has been causing outbreaks since the mid-90s, right? Mostly in the Nipah belt, where the bats are that harbor the virus. And while it is technically possible that it could become a global, so far it doesn't spread very well among people, and that's what you need in order to become a pandemic, right? And so all the pandemics we've seen so far, they emerge, boom, they're already transmissible highly among people. So I don't see it. I don't see it getting to pandemic stage. But I think we need to have a vaccine ready for those places where there are a lot of cases, and if there ever should be a wider outbreak, it would be ready. Oh, David Packman, you misheard a question about pre-party screening. He asked about Abbott Binax rapid antigen. Now, so as I said to Amy earlier, the rapid tests are not great for asymptomatic infections. They're okay for symptomatic, but asymptomatic 50 to 60% accuracy. So no, I don't think pre-party Binax now would be, PCR would be the way to go for sure. Yep. And so I did Packman and Friedman, and I also did the University of Kansas Medical Center podcast last Friday. So I did the rounds, folks. That's probably it for a while. Nobody else is asking me. One in 3 Americans had COVID by the end of 2020. 30% had COVID. What do I think? It's probably an underestimate. Maybe fivefold. Maybe 1 in 5 are not being detected. So I think we're getting very close to having good immunity, but we're not there yet, because only 50-some% have 2 doses of vaccine, right? But perhaps we'll get there. Do cells have many protein-based making apparatuses, or do they make one protein at a time? Oh, they have many. So the protein-making apparatus is a ribosome, right? It's a complex of RNA and proteins, and the mRNA threads through it, and the amino acids get put on, make a growing protein chain, and there are hundreds of those, thousands all over the cell, making all different kinds of proteins as needed. Yeah, not just one at a time. Any comments on the spike protein being cytotoxic? So, I don't know what you mean. Yeah, if you make enough of it, let's say you take cells and culture in the lab, and you produce the spike protein. The cells are going to fuse. That's cytotoxicity. And so, is that a problem in people? No. How many millions of people have gotten mRNA vaccines? I have. My whole family has. They're all fine. It's not an issue. I don't know why he and others keep saying this. It doesn't really play into any issues around the mRNA vaccines. Do some viruses accidentally duplicate their genome and pack both into a capsid? Do you mean put more than one genome in a capsid? So SARS-CoV-2, 2 genomes instead of 1? I think not likely with a virus with such a big genome. I just don't think there's room in the capsid. I think it's pretty full. Influenza virus, though, puts extra segments in for sure. We know that. And so there's certainly other viruses that do so. Naturally, retroviruses put 2 RNA copies in their virus particles. So it happens with some, yeah. Probably not with all, that's all. Oh, Jason, welcome. Glad you caught it live. Yes. If you do it once a week, eventually you're going to catch people, right? There's a huge audience for long-form content. Yes, I agree. Media ignores it because they want to get a lot of content in short with lots of ads, yeah. But we have been doing long-form since 2008 on my podcast. I thought from the beginning it would be cool. We always had 1 to 2 hour shows long before many other people were doing that. I always thought it was valuable, and I still think it's valuable, obviously. Although I think there's a place for short form. For me, short form is 15 minutes. I've made some 15-minute videos that I think can be effective. And I'm going to continue to make short form. That's one of the things I want to do more of in the incubator. Hello from Finland. There's a lot of hoo-baloo in my chess circles about Fauci's lies. Any comment? So I'm not sure what you mean, but I can guess. He told the Senate, months ago that the NIH does not fund gain-of-function research. So I don't think that's true. I think the work that was done by EcoHealth Alliance qualifies as gain-of-function. You change the property of a bad virus, you gave it a different spike, and it does something different in mice. That's gain-of-function. So I don't know why he said that. Maybe he was thinking about some other study. I'm not saying he's lying, but I don't know why he said it, and maybe he was confused. Maybe he's thinking gain of function is gain of function of concern, the subset of gain of function where you make a dangerous virus that can hurt people. Maybe that's what he was thinking of. I don't know. He didn't ask me. Nobody asked. Well, actually, people did ask me yesterday and today because, as you know, the grant application of EcoHealth Alliance has been released by a Freedom of Information Act. And I was sent it by reporters and asked to comment on it. And there's an experiment where they take a coronavirus from bats and they replace the spike with another spike from a different coronavirus from bats. And in mice, it has a different property. It's gain of function. Funded by NIH, done with EcoHealth in China. No question, but harmless. Can you explain why the Delta variant is more transmissible? Well, I choose to say it's more fit than the original. It moves through human populations better. There may be an intrinsic change in the virus that makes it more transmissible. So if you think of a virus going from one person to another, clearly some properties would make it better at doing that. Like if you could make more virus, if it were more stable, and if you made it for longer periods and so forth. We don't know any of that. No experiments have been done to address it. All we do is look at epidemiology, at the spread through the population. And a major part, we had an epidemiologist on a couple of weeks ago on TWIV. He said, movement of viruses through populations is influenced by many factors, not just the virus, which is some part of it, but also human interactions, by ecological, environmental conditions. And so I think two main drivers of spread of Delta have been its fitness. It outcompetes other variants because it probably has immune evasion properties. Those changes in the spike do that. But also because people have been coming together more, as we're going back to more normalcy, more or less. People get together and they transmit it. And so I think it's a combination of all of those things. I don't think it has much of an intrinsic greater transmissibility, though, but that has to be studied in the laboratory. My fully vaccinated daughter, who wears KN95 to school, has been told that she's been exposed to someone with COVID and are mostly unmasked. So you should test her. Get her tested. You can do it in a number of ways and do it every couple of days just to see. When she turns positive, then you should isolate her, and then if she's not at risk, she's probably going to be okay. But if she starts to feel worse, then she needs to get monoclonals. Thank you, Faster Tortoise. Should we use the word data as singular? So technically, the data is a plural word, right? Technically, it's plural. The data are looking good. If you want to say singular, it's datum, which is clunky to me. But people 100% of the time do not pay attention to that. Maybe me and Amy and maybe Kathy Spindler are the only people on the planet who say the data are impressive. And to which people tell me, you're old-fashioned, you don't understand that English changes. No, I understand that English changes. You want to use it the way you want? Fine. I'm going to use it the way I want. And I use it that way because in science we use data a lot, and that's the way I was trained to use the word. But, people can do what they want for sure. Lex Fridman linked individualism with being okay refusing the vaccine. Yeah, I know. I didn't really like that. But I don't like individualism like you can have a gun either. I think that's crazy, but many people in this country do. It's my opinion, right? And he doesn't care that you don't like it, Patricia, so, you know. Fauci is taking the view that Goff requires serial passage. Well, that's wrong. That is absolutely wrong. Genetic manipulation is sufficient to gain a function. I don't know where he's coming from with that. If you know, beer is made with yeast, so is bread. Say you want to make a bread with a different taste, you could modify an enzyme in the yeast and give it a new property. That would be a gain of function. You don't have to pass it. Absolutely 100% wrong. Sorry, Tony, Dr. Fauci. I like Tony, but he's wrong on that one. Why are men more susceptible to moderate to severe? Probably hormonal. You know, the susceptibility by sex varies. In some cases, women are more susceptible to infectious diseases. In some cases, women make better, but in general, women make better antibody responses or immune responses when they're younger. But as they get older, they're not better than men. And it's probably for reproductive reasons, right? It's been evolutionarily selected. But in men, the differences are typically hormonal, but the details have yet to be worked out. Lex interviewed me, dude. Look it up if you want to spend 3.5 hours. Symptoms of long COVID might be CFS and fibromyalgia. So there's overlap. Yeah. Are there any other idiopathies that have viral etiology? I don't think of any offhand. CFS, yeah, and long COVID are the two big ones that jump to my mind. Okay, a report that the authentic mu variant is efficiently neutralized in unspecified cells by sera from people immunized with Pfizer. Okay, that's good. What info will be gleaned from the UK challenge studies? This is such a bad idea to take young people and infect them, and you have the chance of them getting long COVID. Anyway, I think they would like to know, for example, how much virus you need to infect someone with to initiate an infection, how it transmits. You put a couple of people in a room, you infect one of them, how does transmission work? You know, all these fundamental things that we can't know. I'm just worried that the people are going to get sick, and it's not worth it for that information to do that. Why can't we target the nucleocapsid? Well, nucleocapsid will not give rise to neutralizing antibodies. They will not block infection. They will give rise to T cells that will kill infected cells, but that's probably not enough. You probably need a combination of neutralizing antibodies and T cells to give you a good vaccine. And the spike is good in that it induces both neutralizing antibodies and cytotoxic T cells. Thank you, Kevin, for your contribution. New from Lex's podcast. With so many scientific titles, I couldn't tell who has what knowledge or what person I should listen to. Well, you found it for viruses, but remember, I have all these podcasts in different areas, and each of them, they have experts in the areas, because I'm not an expert in any other area except viruses. So you could check those out, too. But welcome. We're glad to have you, and I'm glad that Lex's listeners are broad-minded like that. What about other countries that do not have genome sequencing? Could we use PCR? Yeah, you can amplify any part of the viral genome or human genome and sequence it. I think Amy was saying that. That could be done. Or you can send the samples elsewhere where it can be done. You know, you can put capacity anywhere these days. And during the Ebola outbreak, Western Africa, they didn't have any sequencing capacity. So many labs went there and built sequencing capacity. They brought machines there, gave them to them, trained them how to use them. You can do that in many places. So I think that's great to do that. Can we all be friends if I wait for natural immunity? And please, how will I find out if I have antibodies against it? I don't know what you mean, Andrew. You want to know if you have antibodies as a result of natural infection. You have to get an antibody test that looks for antibodies against the nucleocapsid, because that's not present in the vaccines here in the U.S. What do you say to people that say hydroxychloroquine does work for COVID because it blocks attachment to ACE2? It doesn't. That's not the mechanism, and they don't care. Well, what can I tell you? It doesn't work. It works in cells, in kidney cells in culture, but the virus is infecting your lung cells, and in lung cells, it does not block infection, and the mechanism is not by blocking attachment. So hydroxychloroquine, look, if you want to take ivermectin, you can get it because your doc can prescribe it off-label. It's an FDA-approved drug. I don't really have a problem with that. I have a problem if you take veterinary ivermectin because you can overdose, and there have been many, many reports of people ODing. But hydroxychloroquine does not work for COVID at all, and it can't work, and it can hurt you, and it's hurting a lot of people. And here in New Jersey, in some places, they routinely give it with ivermectin, hydroxychloroquine, and dexamethasone. Sorry, it's crazy. So I don't know what to say. Friedman opened up people to true quality research-based discussion. That's good. He did that long before me, for sure. Right? Have you heard about the efforts to make a vaccine against a bacteriophage? I have not, and I don't know why you'd want to do that. Because they can be good, right? They regulate your microbiome. Have you heard of studies that involve nasal irrigation? Not formal clinical trials. People say this works, but it's only going to be temporary because there'll be a decrease in the viral load, and then it will go back up again as cells continue to produce virus. So I don't think they're going to be of any use whatsoever. Unless you want temporary protection. So I go to the dentist, you gargle, right, for 30 seconds. You decrease viral load, but you're in there for a half hour, and then you're gone. So maybe that helps. But them wearing a mask is better. Oh, he broke it up into smaller segments? I didn't know that. Cool. Very good. Someone broke it up to the part where I said ivermectin was used in New Jersey, and he said, see, that could be used to show that ivermectin is good. I said, I have no control over how people break stuff up. They broke up my interview with Peter Daszak to show that Daszak made the virus, SARS-CoV-2, right? Isn't that crazy? And that was on Fox News one day. Where's a good place to get rapid antigen tests? What do you mean a good place to get them? You can buy them at your drugstore, right? Binax Now is a good two tests for like $30 or something like that. The spike is not cytotoxic in us, as far as we know, in the sense that it does damage. It may be in cells, but it doesn't matter. Okay, we are getting near to when I have to stop at 10 because, oh, look at 766 people. I have to eat dinner and I have to finish editing TWIV, which I like to post at midnight, and I don't want to delay that, so we'll stop at 10. Here we go. We're just down to where Amy is oh my gosh. You guys have so many great questions. I love it. I'm sorry I can't get to them, but one of these days I will stay till the end, till I get through all the questions. I'll see how many people remain. I'll do that one of these nights. Okay? We have 400 likes. Is that the most we ever had? It might be. I don't know. What do you think are some of the main factors responsible for us not enabling cheap rapid at home? I think, as Amy said earlier, we don't have a good high-affinity antibody, and that's the key. And we didn't have it 3 months in, and we don't have it now. It's really hard to do. Thank you, AB, for your contribution. What kind of variant would dodge vaccine effectiveness? Well, it depends what you mean by effectiveness. you can measure effectiveness against mild disease or severe disease and death. Right now, no variant is largely evading severe disease and death, right? Because the efficacy against that, severe disease and death, remains in the high 90s for example, mRNA vaccines. Now, the efficacy against mild disease has decreased as antibody levels decrease. And I think that's also a problem with the variants being not neutralized as effectively, but I'm not sure how that plays into it. So I don't see any variant arising that evades the vaccines because so far they're protecting you. And so, as I said, if you are infected and recovered and you get a one-shot vaccine, you can handle any vaccine that will ever come up, most likely. So I have no worries that the virus is going to outstrip the vaccine at some point. I think those are fallacies and wrong, simply wrong. Reverse transcriptase any day of the week. Well, look, I think it's a very cool enzyme, but I spent my career working on coronaviruses with an RNA-dependent RNA polymerase. So I have to pick that because I know more about it than anything else, I guess. All right, let's see. Let's go through and find some. We're just getting to the part where you guys are defending Amy. That's great. And that was way back when. Let me thank some of your super chats. Thank you, Charlotte. Really appreciate your donation. Yes, you know where you stand with Amy, okay? That's great. It's not like she's fooling you into thinking that she likes you and she doesn't. She'll just tell you. I'm just curious. You need to change out the CO2. So we have tanks of CO2, which we purchase. They're big. Right? They're almost as tall as me. And they feed into our cell culture incubators. And we make the atmosphere 5% CO2, and that combines with the bicarbonate in the cell culture medium to act as a buffer. So it's a bicarbonate-based buffer, because as cells grow, they metabolize, they produce lactic acid, and the medium would get acidic quickly, and that wouldn't be good for the cells. So this buffers it. So 5% CO2 is for the buffering. And so we have 4 tanks connected to our 3 incubators. There's a backup, but as soon as one goes to 0, I change it. And I change it out because it's too dangerous for anybody else to do, including Amy. I don't want her to change it. So that's why I go in. I also change the liquid nitrogen, which is used to keep the cells frozen. It's just a huge tank, about wider than I am and taller than me, full of liquid nitrogen. You attach to your freezer, and it pumps in liquid nitrogen as to keep the cells cold, and it runs out every 2 weeks or so. So I change that as well because you have to make sure you get the bolt right, otherwise it leaks. Now, when I'm gone, I don't know what's going to happen. We'll have to figure something out. I just happened to see that. Tell us more about your friend who had COVID. He went to a New Jersey hospital. And he and his wife had serious COVID. They were both vaccinated, but they had health issues. And they automatically gave them ivermectin, hydroxychloroquine, and dexamethasone. And then a day later, remdesivir. It's just so messed up to do that. I'm sorry. You talk to Daniel Griffin. He's the doc. I'm not a doctor. Okay, talk to Daniel Griffin and ask him what he says about it. He'll be more diplomatic than I am. But that is just not a good thing to do because at least the hydroxychloroquine doesn't work at all and can hurt you. Yeah, many states the hospitals are full of COVID because they're overwhelmed, right? Hospitals only have so much capacity. They don't have infinite capacity. They can't afford to keep infinite capacity. So the pandemic has pushed them. The beds get filled up. People are overworked and so forth. And those are states where many people are not vaccinated. So that's an issue. So people should at least have some consideration for the healthcare workers. But I think they don't think about that. Thank you, Ditching, for your contribution. Appreciate it. So, Richard, what's his name? Richard Ebright at Rutgers, a big anti-gain-of-function guy. He called me a cult, which I thought was derogatory because mostly cults tell people stuff, try to convince people of stuff that's not good, right? And I certainly don't do that. I think if you want to follow me and learn, then that could be a cult. But, cult has a bad implication, I think, most of the time. A lot of good questions here, but come back next week, okay? I have to figure out a way to get... Yeah, University of Kansas Health Center, really good content. They have great docs on, by the way. I'm really impressed with them, and they're going to come on TWIV in the future. The questions are awesome here. You guys are amazing. Thank you, Pamela, for your contribution to our incubator, the new studio. If someone has no antibodies, by the way, they need to get monoclonals, not ivermectin and hydroxychloroquine. Thank you, Holm, for your contribution. Catalin Carrico and Drew Weissman should get the Nobel for the mRNA vaccines. That's right. Oh, good luck going back to school. Good. Good luck, and hope you're safe. Let's see what other thanks. Here we have Mary, Super Chat. Thank you. We really appreciate your contributions. It helps us keep this going. And I don't want to charge people for knowledge. I think knowledge should be free. And so I give mine away. The only thing I don't is the textbook, which is not published by me, but you can bet if I wrote a textbook my own, I'd give it away. Thank you, Kevin. Long COVID, I already answered that some time ago. Looking for super chats. Sandy, thank you so much. Really appreciate your contribution. Bill, thank you. I understand that with no ability to test for optimization, the Pfizer vaccine dose was kept low. Would this explain why a booster might be necessary? No, I don't think so, because I think Pfizer and Moderna are pretty close despite the dosage difference. It could be that a third dose is what you need to round it out. I mean, that's what Fauci said. You say, most of our vaccines are 3 doses. He said, actually, and Amy told me this today, we always planned for there to be 3 doses. I never heard that. That's news to me. I don't think they planned for 3 doses. They're just learning as they go along. Just be honest. I think it could work out. We'll see. I just don't think the science is there so far. Oh, thanks for all the thumbs up. Oh, I wanted to do this one. Any tips on finding, reading academic papers in general? So, my pods all pick great academic papers, and you should go and look at their reading lists and start there. I think that would be a good start. What do I think of DNA plasmid vaccines? I don't think they work very well in humans. They have not worked out for HIV, that's for sure. So I think mRNA is far better. DNA vaccines work in animals, interestingly, but they don't seem to work in paper. Thank you, Bailey, for your contribution. Did you read the Nature paper? Hybrid immunity, yes, absolutely. We've talked about this quite a bit. There's more than one, and that's what Crotty called it yesterday. Hybrid immunity, yep. A lot of good questions. Take them back next week, folks. Mark, thank you for your contribution. They might ask me back. Their listeners seem to enjoy it. So I'll go back anytime for sure. Thank you, H. Sue, for your contribution. Really appreciate it. Lots of people seem to have seen the Kansas appearance. That's great. The more things I do, I think the more people I can hit, and maybe a fraction of them want to come and learn. Transient. Ooh, thank you for your contribution. Been enjoying your updates while babysitting weather overnight since this little piggy went to market. Our first COVID episode. Wow. Good for you. Thank you so much for your contribution. What do you think about deer? Can you get infected? I don't think so. You don't get very close to deer unless you hunt them, and then you could infect yourself that way. But I think the risk is pretty low. But I do think, the deer are infected. That's for sure. And what else out there is infected? That's going to be interesting to find out. Thank you, Patricia, for your contribution. Really appreciate it. I know, folks, that you're waiting for me to wrap this up so you can leave. A few more minutes. Thank you, Patricia. Is there lymphoid tissue in the nasal mucosa? Yeah, there is nasal mucosa-associated lymphoid tissue, NALT. Could that be preventing infection? Well, it would contribute to it, right? It has B cells and T cells, which will not just sense the infection, but will send out cells to combat it. Yeah, for sure. And any more contributions that I have to thank? Ronnie, thank you so much. You rocked on Lex and Pac-Man. Thank you. Really appreciate that so many folks listened. Mr. Ozzycam, hello. Mr. Ozzycam, I will answer your email. It's going to take me a little time. That's why I haven't yet, but tomorrow I'll get to it. I'm sorry. My apologies. Overjoyed, thank you for your contribution. Sean, thank you. VNA cult member here. Yes, the VNA cult. It's a good cult. Rob, thank you so much for your contribution. Yeah, anyone who would call TWIV a cult would equally call every university a cult. Well, some people do, right? Some people think they're teaching poor things. But of course, not always. And there you go. That'll do it for this Q&A with A&V. Thanks, everyone, for joining us tonight. Thanks for your great questions. Please bring them back next week. We'll be back on Wednesday, 8 p.m. Eastern Time. Good night, everybody. Stay safe.